Hi, my name is Rt-Pcr Assignment about introduction to coronavirus and COVID-19. So this is the picture of coronavirus under microscope. It has nucleocapsid protein, envelope glycoprotein, RNA, spike protein, membrane glycoprotein, and lipid bilayer. Dr. Imelda is the one who first found the strain in 1964 in London. Coronavirus is a medium-sized RNA with 30 kb long, is a single-stranded, and has poly A tail. It is capable to undergo genetic recombinant if two viruses infect the same cell at the same time. It has 4 subgroups, which are alpha, beta, gamma, and delta. Throughout the year, the researchers found many other strains of coronavirus such as SARS, NS63, NL, HCoV, and H and HKU1. So this is the full process of outbreak of coronavirus that happened in the world. In 2003, the name is Severe Acute Respiratory Syndrome, or SARS-CoV, as emerged in China. It is a beta coronavirus members. In 2012, the name is Middle East Respiratory Syndrome coronavirus, or called MERS, also a beta coronavirus subgroup. It's detected from an Arabian couple. In 2019, is the latest outbreak that happened in the world. It also initiated in Hunan supermarket in Wuhan, China. The virus name is SARS-CoV-2. The disease name is COVID-19, and it is a beta group. This is how coronavirus can be transmitted from the animal to the human. Researchers found the host of the virus is a bat. When an individual consumes the bat as a food, the human will become a human host. The virus can be transmitted when the human host has direct contact with other people. It can be transmitted through coughing, sneezing, and droplets. This is how our immune system will respond to the coronavirus. So firstly, after infection, type 2 cell will release inflammatory signal that recruit the immune cell and will release the cytokines that cause vasodilation that allow more immune cells to come to the site of injury and exit the capillary. Then the fluid will accumulate inside alveolus and dilute the surfactant that triggers the onset of alveolia, the decreasing gas exchange and increasing the work of breathing. Then the neutrophils are recruited to the site of infection and release reactive oxygen species or ROS to destroy the infected cell. Then the type 1 and type 2 cells are destroyed, leading to the collapse of the alveolus and causing acute respiratory distress syndrome or ARDS. Then, if inflammation becomes severe, the protein-rich fluid can enter the bloodstream and travel elsewhere in the body. That will cause systemic inflammatory response system or SIRS. Last but not least, SIRS may lead to septic shock and multi-organ failure, and the worst will have a fatal consequence. So that's all from me. Thank you.
Very good day to Dr. Tong and friends. My name is Muhammad Ali. So I'm going to present type of topic. First is technique on how to detect COVID-19, and second is the history and development of COVID-19 detecting technique. For technique in detecting COVID, there are two types of technique. First is real-time PCR. Real-time PCR is purposely to detect the presence of viral RNA specific to SARS-CoV-2 through reverse transcription using sample from nasopharyngeal swab. And second technique is rapid test kit. It will use monoclonal antibodies to detect viral antigens using samples such as blood, serum, and plasma. For Rt-Pcr history and development, first it discovered of nucleic acid replication. In 1971, J. Klim and Nobel laureate published a description on a technique that represents the basic principle of a method for nucleic acid replication. The process that is now recognized as PCR. It could be not verified experimentally during the time because the target sequences required primer, which was not discovered on that time yet. It's a discovery. The picture is the picture of Carey Mullis. He assumed that the primers were added to the nature DNA. They will be extended. The extension product will become unwound from their template and be primed again, and then process of extension repeated. However, this was not the case, and DNA had to be heated almost to boiling after each round of synthesis to the nature newly formed double-stranded DNA. As predicted by Clip, the cleaner fragment of DNA polymerase that was being used for synthesis was then inactivated by the high temperature, and so more enzyme was required at the start of each cycle. So that's all from me. Thank you.
Good day to Dr. Tong and my friends. My name is Ahmad Azim, and today I want to talk about the Rt-Pcr. So what is the component of the Rt-Pcr? First, the NTPs, which allow the extension of the DNA primer strands. Second, PCR primer, which serve as the initial foundation for the DNA replication process. Third, DNA polymerase to synthesize the complementary of the DNA strands. Next, DNA template, which is from a double-stranded from a biological sample, salts and buffer to maintain a near natural pH, and lastly water. So these major components are compulsory to perform the Rt-Pcr. So how does the Rt-Pcr works? First, a sample was collected from a body part where the coronavirus gathers, such as person's nose or throat. Then the sample was sent to a laboratory where the RNA is reverse transcribed to DNA by using specific enzyme, where it was later add additional short fragments of DNA which complementary to specific parts of the transcript viral DNA. Next, the mixture of then placed in a Rt-Pcr machine where the machine cycles through temperature that heat and cool to trigger specific chemical reactions. Next, the cycle repeats over and over, and each cycle double the previous amount to continue copying the target section of the viral DNA. So basically, the standard Rt-Pcr setup usually goes through 35 cycles, which means around 35 billion new copies of the sections of viral DNA are created. As the new copies of the viral DNA sections are built, the marker labels attached to the DNA strands and then release a fluorescent dye, which is measured by the machine's computer and presented in real time. So, when the amount goes over a certain level of fluorescence, this confirms that the virus is present in your body. So, that's all from me. Thank you.
I'm Siti Nofarahani, then Tisha Salihin will cover advantage and disadvantage RT-PCR for COVID-19. As such reverse transcriptase PCR, RT-PCR is of great interest today for the detection of SARS coronavirus 2 due to its benefits as a specific and simple qualitative assay. A major advantage of Rt-Pcr assay is that application and analysis are done simultaneously in a closed system to minimize false positive results associated with amplification product contamination. With Rt-Pcr being so common and well known in research and medicine, the technology is already in place to test for COVID-19. Rt-Pcr can detect current infection of disease, allowing medical staff to determine who is thoroughly affected and who is not. Next, for the disadvantage, Rt-PCR relies on capturing and detecting the virus, and so it is possible to miss patients who have cleared virus and recovered from disease. The distribution of virus across the respiratory tract varies between patients, so even if a person is affected, the virus may only be detectable in sputum or nasopharyngeal swab, but not necessarily at both locations at the same time. Rt-PCR for COVID-19 can only tell if a person is currently infected with this particular coronavirus. It can't provide information on other disease or symptoms. However, due to its time-consuming process and high cost, these methods are not commonly used in clinical sample. In clinical detection, the lack of safe and stable external positive control (EPC) could become a serious problem in the diagnosis of coronavirus, and plenty of attention has been focused to address this problem. However, such a problem could be well avoided in Rt-PCR assay, where APC is an important component. That's all.
Hello and hi to Dr. Tong. I am Ahmad Safwan Hakim, so I will be presenting about importance of RT-PCR during COVID-19 pandemic. So RT-PCR is very important for the need of early detection to differentiate COVID-19 from other coronaviruses. They also can be used to characterize COVID-19 and also the main method for detect causative agent of COVID-19. This can be done because of the six component in RT-PCR, which is reverse transcriptase, mRNA, buffer, the NTPs, sequence-specific primers, and also DNA polymerase, which has been discussed by Azim. So the effect of testing on flattening COVID-19 curve, the major widespread testing is effective on tracking and identifying COVID-19 patient, possible close contact to COVID-19 patient, to prevent new cluster of local spread of COVID-19, to identify study possible mutation of the virus. This testing has been proven effective by South Korea, where they increased portable testing area, wide availability of test kits, testing even for the minor contact of COVID-19, resulting to medical staff in Korea were able to conduct at least 50,000 laboratory tests per day as of February 28, 2020. So why use RT-PCR? Because this technique is used to qualitatively study gene expression and can be combined with qPCR to quantify RNA level. So RT-PCR is used in research laboratory to study gene expression, for example in experiment to distinguish exon and intron, and can be used clinically to diagnose genetic disease and monitor RAP therapy. So improvement of RT-qPCR, use aerosol pipette tips and designated word areas and pipette for pre and post application step. They also optimize PCR parameters, perform preliminary experiment to ensure that PCR condition yield only a specific product, and review the minimum information for publication of quantitative real-time PCR experiment. Okay, that's all from me and us. Thank you very much.